Okay, so today in this video I'm going to discuss 15-PGDH, specifically its inhibition and its downstream effects, stimulating PGE2, which is a prostaglandin. And the reason I am discussing 15-PGDH and PGE2 is perhaps because PGE2 at the moment is my favorite pleiotropic protein, and it just exerts a lot of effects as we see on the board. And it's currently undergoing clinical trials, which is why this is another area of interest, so it's discussed like quite frequently across the literature. To just really go up here real fast to provide some overview, we know that when there's no inhibition, when we have 15-PGDH, just at normal physiological concentrations, we observe that PGE2, once again, it's just a prostaglandin, in addition to other prostaglandins like PGD2, etc., it undergoes typically oxidation according to 15-PGDH. So it just takes that and converts it into inactive form. So once it's in this form, as inactive suggests, it can't really exert a physiological effect that is of interest to us. So if we are able to actually inhibit this, say with SW or AAV9, when we use SW, it's a common inhibitor across the literature. It actually takes this and inhibits it globally. So we'll see a global reduction of 15-PGDH, whereas the AAV9, it's a tropic effect or specific to your muscle tissue, okay? So if you use AAV9, you'll only observe the decrease in muscle tissue, but SW is globally. So we commonly observe SW used across more scientific papers. Also of interest is to note that COX-2 is actually the rate-limiting enzyme for the production of PGE2, and COX-2 inhibitors are actually currently or previously explored for, say, conditions like arthritis. So that is interesting because as of now, we observe that we are actually inhibiting this guy, which complicates this process, because if we see we were previously inhibiting this, which gave rise to this. So we are intentionally decreasing PGE2. But now, therapeutically, we actually want to increase it because we observe the therapeutic merit in actually increasing PGE2, which is very strange, but it's cool. Real quickly, just to go back to the actual inhibition of this guy, we see that if we are inhibiting 15-PGDH, remember 15-PGDH, we are actually going to be increasing PGE2 as well as other prostaglandins, and we see that here. So if you're inhibiting this guy, we're going to see an increase in these guys, which is posited to have therapeutic merit. Okay, and briefly to discuss the actual signaling mechanism that cells undergo. So we have the prostaglandin rate. It binds a given tissue, say a muscle or sensory tissue. We're going to say that it's just sensory tissue, okay? So sensory nerve. It's going to bind the cell. It's going to bind specifically an EP4 receptor, and this mechanism was really delineated using elegant knockout experiments, specifically with an MCK knockout, to decrease the expression of EP4. And when we see that there's a decreased expression, so there's no EP4 receptors in a given membrane, PGE2 isn't able to elicit its effect, and we see an increase in the PGE2 concentration, like systemically. So that said, we won't see these positive benefits, this therapeutic merit in, say, PGE2, because it's not able to actually bind, interact with the cell, and elicit its effects. So briefly, real fast, we see that this actually signals through the TGF-beta, the tumor growth factor beta pathway, or the myostatin pathway. The literature kind of gets funny here, I will say, but eventually we see a decrease in the attrogenes, specifically say attrogen 1 and these other proteins. We'll see a decreased expression of them. Okay, and importantly, we see that autophagy remains intact, specifically mitophagy. And we know that autophagy is really critical to maintain cell functionality. So this is still intact, which is very good, considering that this is being explored for therapeutic merit, because we know that we don't want to compromise cell functionality in response to treated with this inhibitor. Also, importantly to note is that 15-PGDH actually increases with age. So in a younger mice, we'll see that there's a low concentration of PGDH. So if we try to inhibit PGDH in a young mice, we won't see any changes systemically in terms of, say, the muscle benefits that we'll discuss in, because once again, there's not a significant enough concentration of 15-PGDH to really exert any noticeable effect. Whereas in aged mice, okay, in aged humans presumably, we will observe an effect because we've accumulated a built-up concentration of 15-PGDH. So inhibiting this is thought therapeutically to restore, say, youthful muscle, potentially mitochondria, etc. But we'll get to that. So to jump over to the response that we observe, we see an increase in muscle mass, size, and strength in response to inhibition of PGDH. In other words, when we have an increased concentration of PGE2 in a given physiological system, we observe an increase in muscle mass, size, and strength. And this was most prominently observed, of course, in mice. Okay, we also observe an increase in PGC1 alpha, which this is an old friend from a previous video I did exploring the positive benefits of exercise, because we know that PGC1 alpha is actually involved in stimulating, say, like the mTOR pathway and whatnot. Specifically, it's going to stimulate biogenesis for mitochondria, so the creation of new mitochondria, increase the mitochondrial activity, and we observed this, or they observed this through, say, the increased activity of specific enzymes involved in the citric acid cycle. So yeah, we observe an increase in mitochondrial functionality when we are administering a given inhibitor, or we are just more generally increasing the concentration of PGE2. Remember, this is a prostaglandin. However, I will note that there was no quantification of, say, glutathione or any other ROS handlers, which might be of concern, but I'm not going to say it is a concern. So to move on, we are going to discuss bone health. Okay, so we have osteogenesis. A concern, a huge red flag, is the cancerous aspect to this, because anytime we see genesis, right, like cells growing, cells proliferating, we think cancer. So this is a huge concern for a lot of people seriously considering this literature. And we also see an increase in the osteoblast activity, but not the osteoclast, which is very interesting because we see that this bone growth process mediated by PGE2 is the response solely of osteoblasts, not the osteoclasts, which are responsible for, say, breaking down of the bone. This process is actually regulated via sensory nerve, and this was investigated in a given research article, which I can link below, not muscle tissue, which is really interesting. So we see that the signals for osteogenesis to occur follow as observed down here. And this provides the perfect segue to go on a mini tangent. So we hear the common cliche, exercise to maintain bone density. What does that mean? What is the mechanism? I don't know. Now we know. Now I know. So we are going to discuss it here. Say a given person, right? They hold weight. They are doing a squat. Okay, we have an osteogenic cell. Okay, a given muscle cell. It's going to be compressed. This is going to stimulate COX-2. Oh, COX-2. Look up here. COX-2. That guy, he was responsible for the rate-limiting enzyme, remember? The production of PGE2. We are stimulating PGE2. This guy is going to then go and bind a sensory nerve, because recall that this process up here is modulated via a sensory nerve interaction, not a muscle cell interaction. It's going to bind PGE2, signaling once again through this EP4 pathway, and that will therefore elicit osteogenesis by decreasing sympathetic tone. Because we know that if we have increased sympathetic tone, we have increased, say, norepinephrine, this is going to actually stimulate catabolic processes, not anabolic processes. So we're going to be breaking down glycogen storage, breaking down muscle tissue, etc., which is very strange to think about. Like, why would our cells want to build in response to experiencing like active friction or compression? I don't know. It's a nice process though for us because we benefit from it, which is why exercise is tremendously beneficial once again. Okay, so we should all be exercising as much as we can, guys. So yes, that there is the mechanism underlying the importance of lifting heavy things, compressing our bones voluntarily. Okay, more recently, a very recent paper that came out, I believe in 2026, out of the Blue Lab at Stanford, that explored the actual co-administration of a 15-PGDH inhibitor, because once again, this is a very of interest molecule at the moment, in addition to a GLP-1R agonist, so like Ozempic, like those types of drugs, right, to help people lose weight. We actually observed that, interestingly, the weight loss is unaffected, though there was no change in, say, someone losing a tremendous amount of muscle mass. However, the muscle regeneration, the muscle force, the muscle strength recovery was all intact, or they observed no calcification upon injury. I should also note this: upon injury, muscles regenerated at a better rate when they're actually co-administered with the inhibition of this guy, right? And once more, the only thing that really like got me was the decrease in muscle mass, because we are still looking for a way to actually like circumvent that negative aspect of using this amazing drug, right? Okay, and finally, to discuss the neuroprotective aspects of 15-PGDH inhibition. Okay, so an increase in PGE2 is pretty esoteric, I will say, but there's a decrease in perioxidation, which pretty much just refers to the oxidation of like your cell membrane fatty acids, which is an unfavorable process. We have astrocyte neuroinflammation. Okay, so a decrease in that, right? So a decrease in neuroinflammation is always a positive sign, because recent literature really suggests that neuroinflammation is the leading cause of, say, neurodegenerative diseases like, say, Alzheimer's disease and whatnot. We see an increase in the blood-brain barrier integrity, okay? And this is evidenced by the decrease in the astrocytic swollen end feet. For the people who like really care about this, like the aficionados out there, I'm gonna put figures on the screen everywhere. Like for this one is gonna be pretty intense, I will say. But yeah, we see a decrease in the astrocytic end feet, which is important for, say, maintaining the actual integrity of the blood-brain barrier. So to prevent stuff from getting into your cerebral tissue that you don't want inside traditionally. To sum up my discussion of the neuroprotective effects, pretty much what the inhibition of PGDH does in the brain is it just maintains the brain structure pretty much. So real briefly, to summarize everything that I've just discussed in this video, we have the inhibition of 15-PGDH. Remember, this is the enzyme responsible for breaking down prostaglandins, okay? Specifically PGE2, because the other prostaglandins were in fact explored, specifically PGD2, but there really wasn't tremendous outcomes that resulted from the inhibition of that itself. Okay, so to summarize real briefly what I've discussed in this video, we have 15-PGDH. The rate-limiting enzyme for breaking down prostaglandins, specifically of interest, is PGE2, and we know that when we inhibit this enzyme that is responsible for oxidizing this prostaglandin, we actually see an increase in PGE2, because if we are decreasing the thing that's going to break it down, we are going to see an increase in the actual precursor itself. In this prostaglandin, PGE2, has actually been observed to be involved in, say, neuroprotection, bone homeostasis, and muscle homeostasis. All this said, inhibitors of PGDH are really fascinating at the moment, and they're showing some serious merit, and therefore I'm eager to continue exploring the literature. And yeah, thank you for watching. Have a nice day. Remember to keep compressing your bones, guys, because this is a natural way to increase the concentration of PGE2. So keep moving, keep moving, keep moving.